Hello everyone. This practical demonstrates how to perform Ouchterlony double diffusion technique. Here we have prepared 1% of agarose gel in 1x assay buffer, which is nothing but the phosphate buffer saline. So we have taken 1 gram of agarose mixed with 100 ml of 1x buffer. Initially the buffer was provided as 10x, so we need to convert 10x buffer to 1x by making 10 times dilution. That is 90 ml of distilled water plus 10 ml of 10x buffer, so that we have 100 ml of 1x buffer. Okay, so in this practical we are taking that 100 ml of 10x 1x buffer and mixed 1 gram of agarose into it and distributed 5 ml in each of the test tubes, and now it is getting heated so that the agarose will be dissolved completely. After complete dissolve of agarose, we need to put this sample into the slide. Slowly the agarose is getting dissolving. The agarose is completely melted here, and now we need to take a clean grease free glass slide. So we have taken a clean grease free glass slide here. We need to put this agarose sample 1% agarose on the surface of the clean grease free glass slide, so that the sample will not pour off from the edge. So we are adding now the agarose gel. So we have poured the agarose gel, and we have to keep this thing for at least 5 to 10 minutes so that the agarose will be solidified. Now the gel is getting solidified after 10 minutes, and this particular thing is the template through which we can make a pour through the gel, and this is the gel puncher. So we can put this template under the gel, and by the help of this gel remover, actually gel punching machine, we can cut the gel accordingly. Okay, so we are now placing the gel puncher here, and now with the help of this sucker we can remove the gel. So after removal, this gel looks like something like that. Okay, so we need to level these things as per the uh, leveling provided by the kit. So this is a high media kit, and according to this thing we have to level our slide accordingly, that antiserum X, antiserum Y and antiserum Z is provided, and we have to add, for antiserum X we have to add two antigens, antigen X1, antigen X2. For antiserum Y we have to add antigen Y1, antigen Y2, and for antiserum Z we have to add antigen Z1 and Z2, and for this we have to use 10 microliter sample. Each of the 10 microliter antiserum X and antigens will be added accordingly. So these are the antigens and antiserums that has been provided by the kit. This is antiserum X. This is antiserum antigen X1, antigen X2. Accordingly, this is antiserum Y. And correspondingly, Y1, Y2 antigens are there. Simultaneously, antiserum Z, antigen Z1 and Z2 is provided. So this is a 10 microliter micropipette. We have set 10.0, so it is actually set it as 10 microliter. So we need to take 10 microliter each of these antiserum and antigens into the corresponding vials. and accordingly we have to add the all the things. After addition of all of these things, we need to put this sample in a moist chamber for 24 hours. After 24 hours of incubation, what we can see in the sample X, antiserum X and antigen X1 and X2, we can see a clear band of identity. That means the two antigens here share some common epitope, so that the antibody that is specific for each of these antigen is actually interacting and ultimately forms a single curved line of identity. However, for the Y sample, Y antiserum and Y1, Y2 antigen, what you can see, there is a partial identity. That means the two antigen, that is Y1 and Y2, share some common epitope. However, one of them has unrelated epitopes. For the Z sample, the clear cross bands, actually precipitation bands actually appeared. That means the Z1, Z2 here had some unrelated antigenic epitopes on their surface. So the antibody, two types of antibody that is present in the antiserum Z is actually interacting with the corresponding antigen wells present in the different Z1 and Z2 sample, and that will cross together. So this is actually the non-identity. So X actually was the identity, Y was actually partial identity, and Z was actually the non-identity. So this is actually the pattern of identity of the Ouchterlony double diffusion technique.